Okay, so we left off with the esophagus, and now we're going to get into the stomach. So this is the point where things get a little bit more complicated, just in that this is where all the action is. So we'll go over, again, anatomy, and talk about secretions, and talk about what part the stomach plays in digestion, and then we'll go from there on to the next lecture. So the stomach is really the first part of chemical digestion. Even though there's some enzymes in saliva, they don't make a lot of progress before they hit the stomach, and they're actually, sorry, my cats are playing quite loudly. The enzymes in saliva are actually inactivated when they hit the acid of the stomach. So what the stomach does, first of all, it's quite expansive, and so it can hold several liters of food plus fluids for temporary storage. This is part of what allows our digestive system to pretty much access all of the nutrients in food, because most of the time nothing has to rush through. We'll talk more about that as we look at the different parts of the stomach. It's also a major mechanical digestive location. There are 3 layers of smooth muscle in the stomach rather than the standard 2 that are in the rest of the system. And this is where protein digestion starts, because the stomach secretes both an enzyme that starts breaking down proteins, as well as hydrochloric acid, which makes it easier for that enzyme to work. This is also the point where we change the name of what we have ingested. So basically, when we mix food with saliva, as well as all of the secretions from the stomach, it becomes something called chyme. That's what we call food mixed with gastric juice. So it's a little bit gross, but we want everything mixed really well together, because otherwise we can't have good access of enzymes to break down all of our nutrients. So when we look at the anatomy of the stomach, we can divide it up into 4 regions that do have different functions, basically. So the cardia is the smallest. It's literally just the small area right around the lower esophageal sphincter. So it's the entryway into the stomach. The fundus is the part of the stomach that actually sticks superior to the cardia. I'll show you on the diagram in a second. The body is what we call the majority of the stomach. And then the pylorus is basically the last part, as in it's got the opening to the small intestine, which is the pyloric sphincter, but it does have a couple of parts to it, which we'll go over a little bit. And it basically functions to make sure that everything has been as mechanically digested as possible before going into the small intestine. Its other job is to make sure that no more is entering the small intestine than the small intestine can handle. So that's kind of where we get to that whole regulation stuff that we'll talk about in a little bit. When we look at the layers, the stomach has the 4 standard layers that we talked about before. So it's got mucosa, submucosa, muscularis, and serosa. But instead of 2 layers of muscle, it has 3. So it's got a longitudinal and a circular, like everything else, and then it also has an oblique, which basically means 3 different directions of muscle sheet, and that just allows it to have a really good range of motion, essentially, which makes it so good at its mechanical digestion. Now, when the stomach isn't fully distended, the mucosa has basically a surface texture to it. We call them rugae or rugae, can't remember off the top of my head, but anyway, they're folds. And basically it just means, like, when the stomach's distended, it doesn't have any wrinkles in it, right? But then when it relaxes, that's how it kind of wrinkles up to be smaller, and that makes sense. So you're gonna have more rugae when the stomach is empty and less as it gets more and more full, but it's very distinctive. And as we'll see, the small intestine and the large intestine also have their own distinctive mucosal folds that are a little bit different. The stomach also has a shape to it, which you've probably seen before, but if not, you'll be very familiar with. So because it's sort of this modified storage sac, it has a lesser curvature here. This is the cardia. This is the esophagus coming down. This is the fundus, the body, and then the pylorus comes out this way to the small intestine. So lesser curvature is here and greater curvature is here because literally it's a really big curve. So here's a nice diagram of that, showing you the different regions. And the pylorus has several different parts to it. So it's got an antrum, which is the first part, a canal, which is where it narrows, and then the sphincter, which is a thickening of the smooth muscle, controlling the exit. And so this works as kind of a filter. So things that are food that's still too large isn't allowed through. And that way the small intestine only receives food particles that are small enough to successfully be digested fully. I don't think I wrote that down anywhere else. Okay, so then when we look at it on a microscopic level, we see that there's several different types of cells. Now, because the stomach literally secretes hydrochloric acid and has an average pH of about 2 to 3, its secretions are damaging to the epithelial cells. So the surface of the mucosa is only lined with one type of cell, which is the mucosa cells that secrete an alkaline mucus. So the alkaline, it's bicarbonate, and it's going to neutralize the hydrochloric acid just at the level of the cell, so that they're not constantly being killed by the gastric acid. We'll talk about that more in a little bit. And then the other types of cells are protected from the secretions by being located in these pits and glands. So basically there's a whole bunch of holes on the surface of the mucosa that are called gastric pits. And most of the epithelial cell types are in these really deep tunnels, and we call them gastric glands. And that's because those cell types are secreting everything that comes to the surface and becomes the gastric juice, which is all of the secretions from the stomach. So the different cell types are named by what they do, or I don't know, some of them just have weird names. So the surface mucus cells secrete the alkaline mucus to protect the mucosa. At the opening of the pits, we have mucus neck cells, and they actually secrete acidic mucus. I don't know why, they just do. Now, what we're going to find throughout the entire GI tract is there's always going to be stem cells or regenerative cells to replace all of the other types of cells, because they don't last very long. In spite of the alkaline mucus in here, they're still going to die fairly frequently. And we see that throughout the tract. This is a high turnover epithelial tissue. The types of cells that we see in the pits are the parietal cells. They're the ones responsible for actually secreting the hydrochloric acid. They also secrete something called intrinsic factor, which is required for the absorption of vitamin B12, which I should have written down somewhere else later, but just in case I don't. And ghrelin, which is one of the like hormones, although not a super important one, or I don't know. Anyway, the key thing here is parietal cells secrete hydrochloric acid. chief cells secrete pepsinogen. Pepsinogen is the inactive form of the protein enzyme. And then they also secrete gastric lipase, which is an enzyme to digest fats. The weird thing about that is we don't think it has a ton of activity, but they secrete it anyways. Now, the last type of cell is actually a group of cells called the enteroendocrine cells. And those are all of the hormone-producing cells. It's just our generic term for the hormone-producing cells in the digestive system or the intestinal tract specifically. So we'll see the small intestine has these guys too. They're different types though. They all make different kinds of hormones relevant to where they are. And these are going to be major regulators of digestion that work alongside those enteric nervous system plexuses that we talked about last time. The big one, really the only one that you need to worry about, is the hormone gastrin, which has major effects for encouraging the stomach to increase its secretions when it's getting filled with food. And it also has interactions with the small intestine that we'll touch on a little bit more later. So all of this mucus and hydrochloric acid and stuff, it has a combination of effects. So the gastric juice, with its really low pH and its literally protein digesting enzymes, is deadly to the epithelial cells lining the stomach. But it's necessary to digest the proteins in what we're eating in our digesta. And it's also necessary to kill the majority of bacteria that we ingest along with our food. Most bacteria don't make it past the stomach because of the hydrochloric acid. So the mucus is one of the major factors in keeping these cells alive for a couple of days. So we call it the mucosal barrier. And it's a combination of that alkaline mucus produced by those mucus cells. There's also a lot of tight junctions between the epithelial cells so that the gastric juice can't squeeze between them and get to the underlying tissues. And then there's the inevitable high turnover rate where, you know, in less than a week, we have all new mucosal cells as they're being killed and replaced. So all of this is protecting the stomach from itself. And when this stuff goes wrong, that's when you get ulcers. And, you know, we know that there's certainly a variety of things that can predispose us to ulcers, but ultimately the major factor is a bacterial infection. And once we figure that out, they became much easier to treat. But yeah, anything that breaks this down can predispose you to ulcers because literally the gastric juice will eat through the layers of the stomach. So this is what it looks like on a diagram. So the surface is all just the mucosal epithelium making that alkaline mucus. And then the gastric pit is basically the openings to the gastric gland. So it's all the same structure. The difference is that the cell types up here are just making mucus, and then the rest of them are down here making hydrochloric acid, pepsinogen, and then various hormones and stuff. That's everything. So to regulate gastric secretion, there's a variety of factors. And it's going to be a combination of neural, hormonal, and local control. So neural controls are the long and short reflexes of the enteric nervous system. There is some amount of influence with these long reflexes from the central nervous system. We'll touch on a couple of those in a second. Hormonal controls, although it's not exclusively gastrin, it is mostly gastrin. So once gastrin secretion increases, we're going to have an increase in enzyme secretion from the chief cells and hydrochloric acid from the parietal cells. Now, there are also hormones in the small intestine, which we'll talk about when we get there, that can then also stimulate hormonal control as well. So there's feedback between those two organs. And then the local control is from the parietal cells, as in how much hydrochloric acid is being produced. And that's basically a matter of if you're putting more food in the stomach, some of that is going to neutralize some of the acid, but the stomach needs to stay at a pH of about 2. So the more stuff goes into the stomach, the more hydrochloric acid needs to be produced by the parietal cells. And so that's going to be controlled by a combination of these other factors. So gastrin, acetylcholine from the parasympathetic nervous system, as well as histamine, excuse me, acting in a hormonal or local fashion, will all increase the binding of hydrochloric acid. And the fact that histamine can do this is how some of our drugs that are for like heartburn and stuff work, because they block those receptors and decrease your hydrochloric acid secretion. All right, so 3 phases of control. It's basically a timeline. So we're talking about temporal phases. So we encourage gastric secretion before we actually eat, or before the food actually gets into our stomach. So that's the cephalic phase. So awareness of the fact that food is coming, and we've all experienced this, we know what this feels like. So seeing food, especially smelling food, knowing that you're getting food, all that is going to stimulate secretions before you even get food in there, so that your stomach is prepared for a bunch of stuff coming in. So that's the cephalic phase. It's basically an anticipation of eating. The gastric phase is how we have increasing secretions when there's food in the stomach. So mostly this is going to be based on like stretch receptors, as well as like chemoreceptors being aware of various elements of what's in the food. And it typically lasts about 3 to 4 hours, because that's how long it takes for a meal to move through the stomach, for the stomach to fully empty after you eat, on average. And then the intestinal phase is how the small intestine has an effect on the stomach's ability to empty, so how fast it's emptying food. So generally this is actually decreasing the motility of the stomach, because basically the duodenum, the first part of the small intestine, when it receives a little bit of chyme, it'll be like, okay, that's enough. That's all we can handle right now. Slow it down. And that like tightens the pyloric sphincter and slows down the motility in the pylorus, so that the intestine is always able to both neutralize the acid in the incoming ingesta and also like actually appropriately digest it, so that we're not losing nutrients through the small intestine. So that's how we regulate gastric secretions in, like a little nutshell. This is the like pretty little flow chart that they made, showing you those things. And this is also nice because it shows you what stimulates in each phase, and it also shows you what inhibits. All right, so a little bit more about what the stomach's actually doing when it's handling food. So this is where we just kind of look at those 6 functions of the overall GI tract, and we see that the stomach is capable of 4 of them. So mechanical digestion, chemical digestion, absorption, and propulsion. And quite frankly, almost all of the tract is capable of these things. It's the other 2 that are special, right? Ingestion is only in the mouth. defecation is only at the anus. So mechanical digestion, we'll talk a little bit about the specific movements that the stomach does. The chemical digestion is mostly the hydrochloric acid and pepsin breaking down proteins. But we must always mention the lipase, even though, trust me, it's really not making any progress on the lipids. Very few things actually absorb directly through the stomach, but there are some exceptions, including alcohol and aspirin. And that's why alcohol is going to hit you really hard if you haven't eaten, because there's no food to mix the alcohol with in the stomach, and it's just all hitting the lumen, or all hitting the mucosa and just absorbing really, really fast. And then propulsion, of course, is going to be linked to the mechanical digestion because it's all a function of the muscular layer, but this is how we're moving the chyme into the small intestine. So the mechanical digestion, it's a couple of different things that go together. First of all, we're trying to mix the incoming food boluses from the esophagus with the gastric juice. And that's how we get chyme. It's just what we call that mixture of things. As I said, the pylorus acts kind of like a filter to control what exits the stomach. So the food particles have to be small enough before they're able to exit. Otherwise, they have to keep staying in the body of the stomach to get broken down. And so gastric emptying is always a very slow process where just a few mils of chyme is actually able to squirt through the pyloric sphincter. So that's under very fine control as far as what volume is allowed out. The chemical digestion is a combination of things here. So the hydrochloric acid is really key. It does a lot of things. First of all, it kills, as I said, most of the bacteria. So this is actually an immune function of the digestive system. Immune function. It activates pepsin. So the pepsin is secreted, as I said, in a form called pepsinogen. I'll talk about this concept a little bit more in the small intestine. So this is an inactive precursor. And it's like that because we don't want to digest the cells that are secreting it. So to keep the cells safe as they produce this protein digesting enzyme, it's secreted in an inactive form. And it's not actually active until it's in the lumen of the stomach with the hydrochloric acid. And the acid itself breaks it down into its active form. And then it's actually able to start breaking down proteins. The third function of the hydrochloric acid is to denature the proteins so pepsin can break them down. So denaturing is when we alter the shape of them, and this allows the pepsin to have better access to the proteins. And then this isn't really chemical digestion, but if I don't put it in somewhere, I forget. So the intrinsic factor, also secreted by those parietal cells, is required for vitamin B12 absorption, as I said. Now, the absorption part doesn't actually happen until the small intestine, but if your stomach is not producing this, then you don't absorb the vitamin B12, and then you suffer the consequences of not having enough, which is generally like anemia and stuff, because you need it for red blood cell production. Okay, I thought I had pictures of the stomach moving stuff. Apparently I did not. That's okay. It's just kind of sloshing things back and forth. Anybody have any questions about the stomach before we get to the next part? Okay, so before we get into the small intestine, I like to do the abdominal accessory organs first, because they are going to be contributing a large number of the secretions into the small intestine for digestive purposes. So to me, it makes more sense to cover them first, and then we go to the small intestine and look at their secretions in combination with what the small intestine is doing. So we're going to talk about the liver, the pancreas, and the gallbladder. So accessory organs, remember, are part of the digestive system, but not part of the gastrointestinal tract. So they're not directly in the tube, but they are contributing to the functions of the system. And so these are the other major accessory organs. Everything else is in or related to your mouth. And their job is to provide really important compounds to aid in the chemical digestion in the small intestine. Basically, we cannot digest things with the small intestinal enzymes alone. We need the products of these organs. So the liver makes bile, and this is necessary for fat digestion. And we'll talk about the digestive parts later. The gallbladder stores excess bile that the liver produces in between meals and then releases it as needed. So we get bile from these guys. And then the pancreas makes a combination of products called pancreatic juice. So you'll notice whenever we have a secretion, we're generally calling it juice. Try not to overthink that one because it makes it hard to drink actual juice later. So this is a combination of enzymes as well as bicarbonate and a whole bunch of water for nutrient digestion. And then the bicarbonate is to neutralize the hydrochloric acid from the stomach. Because the small intestine is not an acidic environment and its enzymes do not work in an acidic environment. So the bicarbonate is easily as important as the enzymes. And then as we'll talk about on Wednesday when we get to the metabolism chapter, the liver also processes everything absorbed by the small intestine into the blood. But we'll get to that part of the liver's functions later. Right now we're just focusing on its exocrine functions and its anatomy. So here are those organs. So you can see the esophagus coming down. The stomach would actually be on top of this stuff, and you can't see it because you had to remove it to see this stuff. But this is the beginning of the duodenum here. So we've got liver, gallbladder, and pancreas all sitting here, and they've got little ducts that secrete right into here. And you'll be learning the anatomy of all of this stuff in your lab sections. We'll talk about that later. Sorry. Okay, so the liver is basically divided into two lobes by a couple of ligaments. And remember we talked about the peritoneum and stuff, and I mentioned like the falciform ligament connecting the liver to the anterior portion of the abdominal wall. So it also divides it into two parts. And there are actually a couple of other lobes, but we're not going to worry about it for lecture. Blood vessels all enter the liver at a specific location that's basically right up the middle in this division. It's called the porta hepatis. A portal is just an entryway, okay? So it just means the liver enter. Liver door? Liver door, that sounds about right. Okay, so we've got a hepatic artery and a hepatic portal vein. Now this is weird because most organs have an artery carrying oxygenated blood in and then a vein carrying waste products out. But the liver has an artery carrying oxygen in and a vein directly connected to the small intestine carrying all the nutrients that are absorbed. So this vein is actually bringing things to the liver, including any toxins that you may have absorbed, and this means any drugs too, because your body doesn't know the difference. There's then an additional vein exiting out the other side of the liver that's actually carrying waste products. So the hepatic portal vein is weird, or I mean, it's super critical, but it's unusual. The other tube going into and out of this location is the common hepatic duct, which carries bile out of the liver towards the small intestine. Now, when we look at the microscopic anatomy of the liver, we find that it's pretty much exclusively made of liver cells, which are called hepatocytes. And they're organized into both structural and functional units. So the structure is the function and the function is the structure. That's why it's together. called liver lobules. So basically we have these sheets of cells, and they're flat sheets of cells that are basically in these hexagons. And there's a central vein running through the middle, and that central vein is leading to the actual exiting vein, which hooks up to the inferior vena cava. It's, I think we just call it the hepatic vein, and that's why this one has a different name. Anyway, and then at each corner of the hexagon is a triad. So a portal triad, which is different from the last time we learned a triad. A portal triad has a branch of the hepatic artery carrying oxygenated blood, a branch of the hepatic portal vein carrying nutrients from the small intestine, as well as a bile duct, which carries bile produced by the liver out. So we have incoming blood from the two different sources, as well as the bile coming out. So what happens is the blood all runs through the lobule, getting processed by all of these hepatocytes, and then draining out of this central vein. And then all of these hepatocytes are making bile that runs in these teeny tiny little ducts called bile canaliculi, and then heads out towards the larger bile ducts. In between these lobular plates are hepatic sinusoids. And so that's basically where all of this blood is in these big, thin venous sinuses for maximum filtration of the blood and before it gets filtered out through that central vein. So that's how we have a structural and functional unit. Basically, all of these hepatocytes are capable of doing all of the same things, and what they actually do on any given day depends on what they're getting in that blood and how much bile they're being stimulated to produce. So the sinusoids are these spaces for blood. They're very porous. So we have fenestrated capillaries. Most capillaries in the body are not fenestrated. They are tightly controlled in what diffuses out. You'll learn about it more when you get to the cardiovascular system. But these guys literally have holes in them. They're very, very leaky. And that's good in the liver because the liver filters everything from the blood. So this gives hepatocytes maximum access to all the nutrients for processing, which again, we'll talk about more on Wednesday, and any toxins and other waste products coming from the intestines. So one of its major jobs is to detoxify the blood that's coming from the intestines. There are also a bunch of phagocytic cells hanging out in these sinusoids. Their name, if you can believe it, is reticuloendothelial cells. And so any like if you get any like bacteria or other like cellular debris, anything like that, they are going to eat that. So the liver literally filters the blood. And then, as I said, all of these hepatic sinusoids, they kind of come together to form those central veins. And then throughout all those lobules, the central veins come together to form the hepatic vein, and that sends the blood back to the heart and the general circulation. But now it's been filtered free of toxins and other things. And so that lets the liver help keep us safe. And what it does with the nutrients, we'll tackle on Wednesday. So here's what that looks like. And we'll cover a little bit of this stuff in lab as well. We're not going to get into like all the details. So the liver makes almost 1 liter of bile a day, which is pretty impressive. Yeah, so it processes all those nutrients, like I said, and detoxifies. It also stores some of the fat-soluble vitamins especially vitamin A, so that if we get more than we need, we have a little backup source. All right, so that takes us to the gallbladder. And the gallbladder is literally just a storage sac. So a bladder is just any, you know, little dead end expansive thing. We have two. We've got a urinary bladder and we've got a gallbladder. Other species have other bladders. So a lot of fish have an air bladder that they can put gas into for their buoyancy. So that's why we have these prefixes. So even though when we usually say bladder, we mean urinary bladder, we've got the gallbladder too. And its job is to store bile. It also concentrates the bile, so it removes a lot of water from what the liver produces, and then it secretes it into the duodenum, the first part of the small intestine, as needed when we eat. So there's three parts to it. There's the body, the fundus, and the neck. It just has a 3-layered wall, so it doesn't have a submucosa. So it just has a simple columnar epithelium for the absorbing of the water. It's got a muscularis for the secretion, and then it's got a serosa because it's in the abdominal cavity. A cystic duct, that's what we call the duct that connects it to the one coming out of the liver, the common hepatic duct, and then they come together to form the common bile duct, which then goes to secrete bile into the intestine. So that looks like this. So from each side of the liver, we have a hepatic duct coming together to form the common hepatic duct, which comes together with this cystic duct to form the common bile duct. And when we're not eating and we're not secreting the bile, there's a sphincter at the end of this basically that closes off. And so then all the bile that the liver makes gets shunted back up into the gallbladder. And that's how the gallbladder receives its stuff. It also has, like, I think they are also called rugae. It has texture to this because it is, again, it's expansive. Now, sometimes it overdoes it and concentrates the bile too much, and that's how we get gallstones, which I'm told are quite painful, which makes sense because they're going to block things. Now, the nice thing is that you can have your gallbladder removed. It's not critical. You just don't store extra bile, but your liver still makes it. So it's fine if you need to lose it. Just sucks before they actually figure that out and take it out. All right, so that takes us to our last accessory organ in the abdomen, which is the pancreas. So the pancreas is actually both an endocrine and an exocrine organ. Its endocrine function is to make several hormones. The one I'm sure you've all heard of is insulin. But the majority of the pancreas is exocrine. So it's producing the pancreatic juice, this combination of enzymes, bicarbonate, and then there's going to be a lot of water in there too. So the exocrine pancreas is exocrine glands. So we have acini, which are secretory cells clustered in little circular things, and they're producing the enzymes and zymogens, which is the fancy term for precursors. Talk about that in a second. And then the ducts, they're not just a pathway out of the pancreas, they also secrete the water and the bicarbonate. So this combination of water, bicarbonate, and enzymes is pancreatic juice. And the pancreas makes enzymes for all 4 categories of organic compounds. So there's proteases for proteins, amylase for carbohydrates, so starch. So amylase, just like what you make in your saliva, there's more of it coming from here. Lipases to get lipids, and then nucleases to get nucleic acids. So we actually do digest DNA and RNA and absorb the monomers. So it kind of looks like this. The pancreas is this weird little taily looking thing with the pancreatic duct running through it. and it joins up with that common bile duct right before they actually both secrete into the duodenum. We'll tackle all that stuff later. These are the acini of the exocrine pancreas and then the ducts. And then there's these little islands of endocrine tissue in there as well, literally called islets, you know, like tiny island. Pancreatic islets is the newer one. Islets of Langerhans is the old term, so you might see that too. So getting the stuff into the small intestine, for whatever reason, I guess for efficiency, like I just showed you, they come together, the bile and the pancreatic juice, and so that combined outlet in the small intestine is called the hepatopancreatic ampulla. And it's got a little smooth muscle sphincter right there, the hepatopancreatic sphincter, that controls when things are actually secreting. And then sometimes there's a second duct straight from the pancreas, but I guess it's variable, and so we don't really consider it to be a critical thing. So here's a nice diagram showing all of that stuff. So you can see common hepatic duct again, cystic duct, common bile duct. There's a little sphincter down here. And when that's closed, we're going to get bile going back into the gallbladder. Pancreatic duct, little sphincter here, comes together as this ampulla. And there's a little valvy guy here that is the sphincter down there as well. It technically sticks out into the duodenum, and we'll see that in lab. So the major duodenal papilla is just literally the visible bump of the opening of this ampulla. And I think that little hole there is meant to be that secondary one, but you don't have to worry about it. And this one's nice because it gives you the new names, but it also gives you the old names. So they actually called it the ampulla of Vader. Spelled differently from Star Wars, but still. It's a good thing we changed all these things. Okay, so you'll learn more about that. Well, not more about that, but you'll get more familiar with that in lab. Why did I put this here? I don't remember. This is our overview. We'll hit it again later. Didn't I have zymogens in here somewhere? Nope. Okay. I forgot to take these two out. Sorry about that. Just ignore these two. They're actually meant to be at the end. They are at the end, but they're also here. Okay. I guess I put the zymogen one in the intestines. Sorry. We'll tackle this whole precursor enzyme thing in a little bit. Anybody have any questions so far? All right, give me 1 second. Let me see what I did. Nope. Okay, that makes sense. Cool. This is why I'm getting confused, because I went and re-edited it and put it in the next one too. So there's a lot of overlap, apparently. Sorry about that. It's been a weird week. Before I totally finish that one up, let me go back and just mention, there it is. This slide, which we should have tackled before. Bile, what is bile? I didn't have that in there, and I made a slide, and it's actually on the next lecture because I got confused. So bile is an alkaline solution. It contains bile salts, bile pigments, cholesterol, triglycerides, phospholipids, and electrolytes. So it is made for the digestion of lipids, okay? And it actually contains lipids in it. Now, bile salts are actually made from cholesterol. Bilirubin is the most common one, and they are amphipathic. So very much like phospholipids, remember how they have a polar head and then they've got the non-polar tail, so that they can interact with polar molecules like water on one side and non-polar things like other lipids on the other. The bile pigments are actually waste products from the breakdown of red blood cells. So the heme part of hemoglobin is the primary thing that makes up the bile pigments. So it's a waste product that we kind of use as part of our fat digestion. Now the bile salts actually get heavily recycled. So there's something called enterohepatic circulation. So if something is secreted in the bile, it'll actually get reabsorbed at the end of the small intestine, so the ileum. And then as we just briefly talked about already, everything from the intestine goes by the hepatic portal vein into the liver to get processed. So then the hepatocytes will pull out the bile salts and secrete them again. So enterohepatic circulation is how we recycle most of our bile salts. And so a little bit of it ends up out in the feces, like we do lose some, and we do always have to make new stuff, but for the most part, we're actually just recycling it over and over again. Okay, so here's a little picture of that. So this is obviously kind of a vague diagram. So the intestines here, this is actually the liver. I know it doesn't look like the liver, but it is. And so this is bile secretion. This is the portal vein and bile being reabsorbed. And then this is a little bit of bile actually ending up in regular circulation, which is relevant later. So the bilirubin, as I said, is from hemoglobin breakdown. This is why bile is actually green. It doesn't always look green because it's very, very concentrated a lot of the time, so it kind of looks more of like a dark brown, but it's actually mostly green. But when it gets to your large intestine, the part that doesn't get recycled, the bacteria break it down into another pigment called stercobilin, and that actually is why poop is brown. So that's always my fun digestive system fact. And then the one that escapes, the little bits of bile salts that escape into general circulation, they get dealt with by the kidneys, and they get converted into another pigment, and that one makes your urine yellow. So a lot of the colors in our life are due to heme products. It's pretty much that and melanin. That's it. So yeah, now you know why different things are different colors. Oh, that sparked questions. Okay, so this is from the second PowerPoint. So or not the second one, but the second digestive system PowerPoint. So basically, I thought that I only had stomach left to do on the old one, and then proceeded to continue to edit the rest of it. I don't know what happened. So it's posted, but it's 21, and I'm going to skip a little bit because some of it is like this slide, for example, is we already saw that, but because I threw in those bile ones, I wanted to touch on that. Like we talked about this already, and there's that, and the Gall Stones and the pancreas. But I found this nice diagram, for example, of pancreas to kind of show you a little bit more about the secretions. And then I knew I had this. This is why I was confused. So zymogens, I said they secrete zymogens. So zymogen is the overall term for a pre-enzyme. So just like pepsinogen is secreted into the stomach and then the hydrochloric acid activates it, So we have the same thing with the pancreatic enzymes, specifically the protein digesting ones. So the rest of them aren't secreted as zymogens. They're already active and ready to go because the cells don't really have to worry about like amylase breaking down stuff in their own cells. But protein digestion is always really scary. So protein enzymes are always secreted in an inactive form. And so the general term for that is zymogen. And they won't be activated until they're in the small intestine. So it actually requires an enzyme in the small intestine attached to the brush border, so the microvilli of the enterocytes, the epithelial cells of the small intestine. So enteropeptidase, or in this diagram it's enterokinase, it doesn't matter, is the one that activates all of the peptidases, the protein digesting enzymes. So basically this one, trypsinogen, is the zymogen for trypsin. It gets activated by enteropeptidase, and then trypsin actually activates the other two, and you don't really need to know the names of the other two, but basically these are then the three enzymes that will continue to break down proteins, basically continuing the job that pepsin started in the stomach. So I knew I had that in there. And then I added a little bit to this secretion one as far as the control of it goes. So the control of secretions is regulated again by a combination of hormones and the nervous system. And specifically those hormones are from the small intestine. So we'll kind of tackle it a little bit more in a minute. But secretin is released by enteroendocrine cells in the small intestine. Endocrine. And it's triggered by the presence of acid. So when gastric secretions go into the small intestine, and the hydrochloric acid will cause the release of secretin, and that stimulates the bicarbonate secretion from the pancreas to make sure we're neutralizing the acid. And then when there's proteins and fats in the chyme coming from the stomach, it triggers the release of CCK. Do not worry about what it stands for. It's a longish word. You can look it up if you want. And that stimulates the enzyme secretions from the pancreas, as well as further enhances bicarbonate secretion. So basically the idea is, you know, there's stuff coming in and we need more juice, okay? And that's how that gets regulated, and then it's gonna get squeezed in to the small intestine. I found this nice diagram that sums that up quite well. Can't even drink my coffee without spilling it. Sounds about right, it's fine. And this one is just kind of showing the pH stuff, but don't worry about it too much. If that looks scary, just skip it. All right, there's the duct one. Whoa, what just happened? Okay, so that takes us to the intestines. So yeah, no, this is intestines, but it's 21. So I edited a little bit more just to kind of streamline everything from what was on the lecture 20 post. So just don't worry about lecture 20 past the stomach, because that's apparently where I thought it ended. Okay, so the intestines. So now we're going to tackle the majority of the length of the digestive system and look at the different parts and the different unique features, and go into the digestion and absorption in the small intestine, and then we'll probably do the large intestine on Wednesday before we tackle metabolism, because it's already 9:30. So the small intestine. The small intestine is the longest part of the gastrointestinal tract. It's about 3 meters. Now the length varies because when you're alive, your smooth muscle tone keeps it all a little bit shorter. And then when you die, it all relaxes and you get like a extra meter out of the length. It doesn't really matter. We divide it up into 3 parts. They all have different functional features and different things happen in them. So the duodenum or duodenum, depending on what region you're from, is the first part. It's the shortest, but it's also really important because that's where all those secretions come in. So it's only about 10 inches. And then the rest of it is jejunum and then ileum. And the small intestine is where digestion basically finishes. Like if it doesn't get digested in the small intestine, it's not getting digested. And it's also where almost all of our nutrients are absorbed. So again, if it doesn't get absorbed here, we lose it because the only thing that the large intestine can absorb is electrolytes and water. Oh, and a couple of vitamins. So this is all small intestine. They made the duodenum purple, jejunum in blue, and then the ileum in pink. And we'll look at all of this more in lab as well. This is showing you with majority of it removed because the duodenum, the vast majority of it is actually retroperitoneal. So the stomach would be right here and the liver's up here. And then this is basically the pyloric sphincter. The duodenum goes retroperitoneal, comes back around, and then the jejunum comes off and does that crazy squiggly thing from there. And you can see that this is where the portal vein goes up to the liver and where the bile ducts and stuff come out. And then here's the pancreas, also retroperitoneal, and its duct is going in here. When I find really cool diagrams, I like to share them. This is a really nice one of the duodenal ducts. So you're getting a better view of the papilla. There's a major papilla, which is where that guy comes in, and then the minor one right here. You can see it's not pictured, that hepatopancreatic ampulla, because it's behind there. All right, so the same 4 layers as everything else. Now the mucosa has a lot of features that massively increase the surface area. So not only is this the longest part, but it also has a huge surface area. Like if you actually laid out the mucosa flat, it would be bigger than your skin, okay? And that's because we want to maximize absorption. So they come in 3 different phases, I don't know. Anyway, 3 different features. So there are circular folds. These are visible folds. Like if you looked at small intestine, you'd be able to see these guys. They're also called plica. And they're basically the same thing as the rugae in the stomach. They're ridges of mucosa and submucosa. And they serve to actually spiral the luminal contents through the small intestine. So basically they help control the flow of everything as it comes through, making sure that everything is contacting the mucosal surface because that's going to be necessary for digestion and absorption. When you look microscopically, you'll find that the mucosa is also in these villi. So villi are hair-like projections. To the naked eye, it just makes it look fuzzy. But when we look at it under a microscope, we see that it's literally these little fingers. That's the epithelium, and that's a little bit of submucosa coming up through. And then these epithelial cells are columnar with microvilli. So the microvilli themselves also contribute a lot of surface area, because remember, that's their whole point, right? Is to increase surface area. And in this case, it's for absorption. We call it the brush border because it just looks fuzzy on top of the cells. I guess kind of like the ground looks fuzzy when there's a bunch of underbrush that needs clearing out. I don't know. Anyway, so the whole point here is increasing surface area. And so this is a nice diagram showing you those different layers. So you can see the plica in here, and then these are showing you the villi. So the villi are these little fingers coming off of, this is one of the plica, okay? And then when you look at each villi, you see that it is lined by all these cells with microvilli, okay? So those are our three surface area increasing features of the small intestine. Here's just a different version of this, basically the same thing. When we look at the histology, we see that there's actually even more different types of cells in the small intestine than there is in the stomach. But most of them are enterocytes. So the enterocytes are the simple columnar epithelium with the microvilli. They secrete intestinal juice, so they secrete watery stuff as well with enzymes. But really, mostly what they're doing is absorbing nutrients. Now, there's going to be mucus secretion here as well. There's always mucus. It just helps everything move. And so that's going to be by goblet cells. Hopefully you remember that from histology. There's various types of enteroendocrine cells in here as well, secreting hormones. And then there's also an immune presence. So Panath cells contribute to immune control. They are deep in the bottoms. So the thing with the villi is they go up, but then they also go down. So like this would be the surface, but the cells have sticky uppy bits, and then they have deep parts. And the deep parts are called crypts. And at the base of the crypts are these Panath cells, and they will secrete antimicrobial agents to make sure that we're not getting overrun by bacteria. There really shouldn't be a lot of bacteria at this part of the intestine. Just like in the stomach, we have stem cells because we have to keep replacing these enterocytes. They work really hard. They don't last that long. And so there's a fairly high turnover, not quite as high, I think, as in the stomach because there's no constant hydrochloric acid, but it's still pretty high turnover here. Hard to find a really good one, so I did the best I could. But you can see we've got our columnar enterocytes. We've got goblet cells secreting mucus, Panath cells secreting various antimicrobial products to control bacteria that are in the lumen. And this one I pulled from somewhere that's clearly focusing on the immune system because it's got some additional immune cells monitoring things too. In addition to all of that, we have this immune cell presence in the submucosa. I think I mentioned it before, but anyway, it's mucosal mucosa. I can do this. Associated lymphoid tissue. So it's part of the immune system. In the small intestine, specifically in the ileum, they are called Peyer's patches. We'll look at them in lab. And their job is to monitor the small intestine, make sure nothing is invading from the lumen. And then if there's too many bacteria or something, they will secrete a protein called IgA, which will bind and inactivate bacteria and other pathogens. The other thing that we see in the submucosa are duodenal glands. Now this is only in the duodenum. They're also called Brunner's glands. And they secrete an alkaline mucus. So they're only needed in the duodenum because that's where we need to potentially neutralize acid. So this is going to supplement the alkaline effects of the pancreatic juice. And these two features are actually how we can distinguish different parts of the small intestine from each other under histology, but we'll tackle that more in lab. So here's a picture of the duodenum with the submucosal glands here. This is all the villi and the microvilli. This is actually stomach because this is right at the junction. And then when we look at what the small intestine does, again, there's the physical or mechanical digestion, and then there's the chemical digestion. It on average takes 3 to 6 hours for a meal to move through the small intestine and be fully digested and absorbed. The intestine uses the small intestine uses two major movement patterns to help accomplish that goal. So the mechanical digestion is going to occur with segmentation. Now the particles are pretty well broken down at this point, so mostly what we need to do is mix all of these components together. So we want to mix the chyme with the bile, the pancreatic juice, and the intestinal juice, and that segmentation is how we do it. So if you remember, I showed you a diagram of that before, and basically we just have different parts of the circular muscle like squishing back and forth, and it just kind of sloshes everything together and mixes it up really well. In order to actually move a meal through the whole intestine, we're going to use something called a migrating motor complex, which is basically just a couple of waves of peristalsis that literally start at the duodenum and work their way all the way through. And this is generally what clears out the small intestine in between meals, triggered by the release of a hormone called motilin from the duodenum, usually triggered by another meal starting. We'll tackle this more when we get to the last part of the system as well, because it all works together. So here's segmentation, sloshy back and forth. And that's of course going to enhance our chemical digestion. So chemical digestion, like I said, this is the last part of the system that does it. So it has to be completed in the small intestine. If it's not, then we do not get those nutrients. They're going to get lost. At this point, carbohydrates and proteins are partially digested. Lipids, honestly, are undigested. Even though we had lingual lipase and we had gastric lipase, it hasn't really accessed it a lot. And we'll talk about why that is, I guess on Wednesday. So like I said, it starts with all the mixing. So the segmentation is going to mix everything together. The first thing we have to do is neutralize that hydrochloric acid, because nothing else is going to work if we don't do that. Otherwise, we're going to damage the epithelium of the duodenum, and none of the enzymes from the pancreas or in the intestines are going to work in an acidic environment. So we have to get that neutralized first, and that's going to allow the chemical digestion to take place. It basically happens in 2 stages. So the pancreatic enzymes keep going from where the stomach stops, and they're breaking down basically organic polymers. So we still have these really long chains of organic molecules, and the pancreatic enzymes have to break them down. The last stage is going to be by the intestinal enzymes themselves, so enzymes made by the enterocytes. They're actually usually attached to the brush border, so attached to the microvilli, and they will break down all these organic molecules into monomers and dimers, because that's how small they have to be absorbed. Now, the reason why they're attached to the brush border is because then as soon as they're broken down, they're already right on outside of those intestinal cells to be brought in. So that just makes sense. Okay, let's just do carbohydrates, and then I guess we'll call it good. So carbohydrates. Pancreatic amylase is going to keep breaking down starch. So salivary amylase kind of like starts things off, but it doesn't get very far before we end up with it in the stomach and it gets inactivated. So pancreatic amylase is going to break down starch into oligosaccharides, so shorter little lengths. And then at the brush border, we're going to break all of that down ultimately into monosaccharides. So we actually have a specific enzyme for each of the disaccharides that we routinely ingest. So maltase is 2 glucoses, sucrase, or maltase breaks down maltose, sucrase breaks down sucrose, and lactase breaks down lactose. The monosaccharides are actually able to be absorbed. So that's glucose, fructose, and galactose. We did cover all of those at the very, very beginning of the class. This is a nice little flow chart of how that works. And so this is the level at which they can be absorbed. How that happens, we'll tackle on Wednesday. Okay, so, okay, I guess I can make it through this part and then we'll tackle absorption on Wednesday. So then with the proteins, again, pepsin got it started. The pancreatic enzymes break the proteins down further. So the pepsin just kind of splits the proteins into smaller polypeptides, but they're still really big. The pancreatic ones, the trypsin and the other guys, they break them down even smaller. And then the brush border enzymes break them down into individual amino acids for the most part so that they can actually be absorbed. Hopefully you can see a theme here. So this is protein digestion in a nutshell using the diagrams from your book. It's going to cover where this stuff happens, and then this is the steps. So the pancreatic ones do it here, and then the intestinal enzymes get this part. So we'll start with lipids on Wednesday. This is a little bit more complicated. It's not as simple as just breaking them down. So we'll pick this up on Wednesday. If you guys give me like 5 minutes, we'll do a pre-lab for your digestive system stuff. I'll show you what we're covering. Everything we're going to cover for lab for the digestive system, I'm giving you right now, so there won't be anything else added on to that, okay? So let me stop this so it's a separate recording. Go get a little more of a drink so that I'm not so tongue-tied, and then we'll do a quick pre-lab.